Hi there, Miss White here. This is a continuation of the lecture on regulation of the cell cycle and cancer. In the last video, we left off on question 16 in your lecture packet, so we're going to pick it up there. Hopefully you had a chance to think it through and look over this diagram again and realize that right here is where the spike occurs, where we get a change from production of cyclin to breakdown of cyclin, and that triggers movement past G2 and into mitosis. So that is going to be this G2 checkpoint that we are able to get past. So just to summarize what we were talking about in the last video, cyclin is a molecule that will bind to CDK, which is known as cyclin-dependent kinase. And it once these bind together, they become a mitosis or maturation promoting factor that is able to phosphorylate other molecules and provide energy to carry on the cell cycle to move on to the next phase. So an important thing to remember is that the cyclin portion of the complex is continually being degraded. So and that was that shape of the graph that we saw, right? During interphase, it would increase, then it would decrease. Well, as you can see in this diagram, there are actually many types of cyclin. There's a G2M phase cyclin, there's a G1S phase cyclin, and all of these are going to follow that same pattern where the amount of cyclin will increase, it will bind to the CDK, and then that complex will then trigger the degradation of the cyclin. So we see that the cyclin then gets broken down, it becomes degraded, and it's broken up into parts and can be recycled. However, the kinase stays around and is continually available to bind to new cyclin that is created. So what if cyclin was always available? As you can see here, I'm letting you know that that effect would be that the CDK right here would always remain active. If it's always active, it can always be allowing the cell to keep moving through the cell cycle. So it is very important for regulation that the cyclin does get degraded in order to allow the cell to slow down at those checkpoints and monitor the intra and extracellular conditions and make sure they're right. Again, the answer for number 18 is very similar. How might a cell be affected by the development of a degradation-resistant cyclin mutant? Again, if cyclin is not being degraded, that means it's not being broken down, which means it's staying bound to CDK. CDK remains active, and the cell can keep going through the cell cycle, maybe even if it is not supposed to. Okay, so we're going to be talking about some different external factors that influence cell division. One that we'll hear a lot about is a growth factor. So you can already guess it's probably going to stimulate growth. But important to realize that that would be a protein, that's released by a neighboring cell that when received is going to stimulate that neighboring cell to divide. So the other part of there being a growth factor is that there has to be a receptor protein that's going to receive it. So there's really two proteins involved, the actual chemical signal and then the receiver of the signal, the receptor protein. If they fit, remember proteins all have a unique shape. If they fit, it's going to trigger this domino effect of chemical reactions that would then cause the cell to move forward through the cell cycle. So a growth factor will stimulate growth. On the other hand, there are going to be signals that would inhibit cell division. It's known as often density-dependent inhibition, means basically that if a cell senses that there's not enough room, it will stop dividing because that means there's not enough resources. Normal cells will do that. They will only grow in what's called a monolayer. However, if cells are, if they contain genetic mutations in which the receptors are maybe defective and they can't tell that they are too close to each other, you end up with a situation where cells will start to pile up on top of each other. And again, that can lead to problems because these cells that are not following the rules and dividing when they are not supposed to are going to be stealing the resources from the healthy cells nearby. And here's another image of that, again showing that normal cells typically grow in a monolayer, a single layer. However, cells that don't respond to those signals to stop are going to continue to grow up in clumps, which is the beginning of a cancerous tumor. So I'm going to stop here, and in the next video, we are going to look at a few learning objectives specifically related to cancer. So please see the next video for the remainder of this lecture packet.